Walking Stick Library presents The Genetic Ledger and DNA Computing with your librarian hostess with the mostest, Eileen Kramer. And don't forget, you can catch this stream live on Twitch.
Okay, this is Todd. He's on Rumble because it's easier sometimes to get to the videos that way. And he found an article. And he's going to talk about it.
Yeah, and this is so robust, by the way, that the IEEE has already developed standards on how it is they're going to measure, track, trace, and create a ledger, pluses and minuses of your biorhythms so that they can harvest those, use a cloud computing node, use a battery as a power source. It's all there. In order to develop international standards, it means that they've necessarily been at this for a very long time, decades, in fact, because it takes a huge amount of data to do this. But those of you that have been watching are aware that website inpersona. Yeah, and this is so robust, by the way, that the IEEE has already developed standards on how it is they're going to measure, track, trace, and create a ledger, pluses and minuses of your biorhythms so that they can harvest those, use a cloud computing node, use.
Okay, I'm going to freeze this right here. This doesn't say anything about biorhythms. Anybody can be looking at the title. Genetic Ledger, pioneering secure and authentic data storage through DNA and blockchain. Okay. That has nothing to do with biorhythms. And it doesn't say anything about my DNA. Okay, what we want to do is a known item search. And we're going to do it in IEEE. That's better. And we're going to make this big today. And what we're looking for is genetic ledger. And that is a, why can't I get the whole word when I want it? That's better. It's a phrase. And we're going to go to IEEExplore because it doesn't always communicate well with the Discover box at GSU. And this is an electrical engineering, I think it's International Electric and Electronic Equipment. It's an electrical engineering organization. So let's get Google. Let's go to GSU Library. Let's let ourselves in. And let's skip this big box and go down to the alphabet. We're going to pick I for IEEE, scroll down, and let ourselves in. And all we have to do, you go over here, is put in genetic ledger, in quotes. There's our article right there. Same article that Todd brought up. We can even get the PDF. Now this is a conference paper. It was at the International Conference on AI and Digital Economy 2024. And here is the abstract. The rapid increase in digital data necessitates advanced storage solutions. This paper introduces the DNA chain system, an innovative integration of DNA data storage and blockchain technology. Our hybrid system utilizes high-density storage capability of synthetic DNA to handle massive amounts of data, alongside blockchain's most robust security and traceability features. We detail techniques for encoding digital data into DNA sequences and addressing compression, redundancy, error correction, multi-layer encoding, organizes data efficiently while optimized polymerase chain reaction processes facilitate swift data replication and retrieval. Live data mutation checks ensure integrity against natural alterations, and self-evolving blockchain technology adapts to new security challenges, enhancing protection against tampering. Okay, I don't see anything about taking anything from anybody. Just data of some kind. And they're using synthetic DNA. They're not using my DNA, your DNA, the DNA of the researcher, whomever. They made the DNA in a petri dish. And if you get to the materials and methods, here we have genesis of DNA, digital twins, and guarding against mutations. One of the primary innovations germinating from our design is DNA digital twins concept. Imagine a vast library where every book, DNA encoded data, there exists a handy catalog card, a digital twin. Well, that's what that is. These cards allow swift referencing of rudimentary data interaction, bypassing the need to dive deep into comprehensive tomes. Such a system provides rapid accessibility, blah, blah, blah. Okay. So, I'm confused. They are using DNA as a storage mode. But I'm finding something really weird about this. Okay, when you have an electronic computer, you have a semiconductor. Looks like this. I can draw one. And it has an on and an off. How about this guy? It has an on and an off switch. I can make the on red, and I can make the off green. I don't know if it's that color in real life. I just like it that color, so I'm putting it in that color. And it's either on or off. And the on and the off are, on is 1, off is 2, off is 0. This is base 2. You only get 1s and 0s. And that's how your computer codes things. Now there's a problem here because DNA has 4 different base pairs. A, T, G, C. Oops. C, and these can be in permutation so that each side so we have A, G, T, C. So you have 4 choices. So you don't just have on and off. You'd have what? You'd have 0, 1, 2, 3. That's different. And how would you build a computer with DNA? Sometimes you realize you don't know what you're doing. Because I sure as heck don't. So, I'm going to check to see if anybody is in the house watching me draw. No, nobody saw me. Okay, so I think it's time for a search. And we're going to use our usual lookup source. And for technical topics, Wikipedia is tough as nails, which is fine. because I'm a big girl and I want my text served up nice and straight. I want my circles the same size. So what we're going to look for is, I'm going to bring this down to 48, DNA. My throat is like really dry. Yes, it's my chest, not my throat, and I'm very hoarse. We're going to look for that in Wikipedia. This is a semi-known item search. Most known item searches are very semi. And we can color that in if we'd like. It's going to be green. Sloppy, 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 sloppy tonight. Sometimes you're just too sloppy for words. And let's go run that in Google. So I'm going to go get me a Google, bring it over, and we're going to run the Wikipedia and DNA computing because the and is commutative. You can run it on either side. Okay, there's your DNA molecule, and it's much nicer than pure silicon, all doped up with stuff. You can make DNA in a petri dish. It uses a lot less energy. And down here it explains, finally down here it's going to explain the methods. And there are multiple methods for building a computing device based on DNA with its own advantages and disadvantages. Most of these build basic logic gates and or not. Yep. So that is, I can show you what that looks like. Some of the different bases include DNA enzymes and nucleotides, and there are different methods. But basically, what they're doing is taking the DNA. They are not using the 4 bases inside it, but they are using it like a semiconductor to do getting me to draw tonight. I like to draw. Okay. This is off or 0. And sometimes they hook them by one thing, but I'm just showing you how they'd hook them by 2. And this would be on. 1. And then you can take combinations of them. So if they are both on, didn't do a good job. Let me try it again. And you have another one that is on, like this. You have an or. Either one, you still get where you're going. If you have, actually that's an and. That's an or, 2 open. One is, an and is where you have, this is an and, not an or. Me bad. I think that's an and. That's an and. So with computers, it's different than the sets. And if you have one open and one closed, here's an open one and a closed one. That's an or, and a not is both of them open. And they open and close in different ways, but I'm not drawing them both like that. This is a not. Don't go in there. And you've got, and so they do it by opening and closing the DNAs. They don't do it with the 4 bases inside it. And you can get DNA anywhere. Swab your cheek, go get some grass outside, spit in a tube. It's not your biorhythms. So this is basically how they do it, and it kind of explains it. The most fundamental DNA computing and molecular programming is the strand displacement mechanism. Currently, there are two ways to perform strand displacement. There is toe-hold exchange and polymerase-based strand displacement. Besides simple strand displacement schemes, DNA computers have also been constructed using the concept of a toe-hold exchange. In this system, an input DNA strand binds to the sticky end or toe-hold of another DNA molecule, which allows it to displace another strand segment from the molecule. This allows the creation of molecular logic components and signal amplifiers, which can be linked into arbitrarily large computers. This class of DNA computing does not require enzymes or other chemicals. Okay. So I kind of get what they're doing. They're not doing base 4, they're doing base 2. Okay. And I may not have drawn these right. I don't know that much about computers. I do know what on and off looks like. So that's what, and these guys are using DNA data storage instead of semiconductor data storage and building a blockchain, and they're using synthetic DNA. Somebody should have at least read the abstract of the article before they posted it. Sloppy, 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 Todd. Thank you for watching. And remember, you can catch me live on Twitch.